Once you give
start live, you tell them within 2 minutes we will start like that.
Yeah.
Can you give me 2 minutes?
No, sir. I will. Once we start live, then we cannot stop. So once you come back, that I will start live and I will tell. Now we will start.
Okay. Right now, you whenever you are free, you can start introducing the speaker. By the time I will be ready. Okay? Now, from now onwards, I am ready.
Okay, sir. I will start. Then I will introduce. Within 2-3 minutes, you have to speak.
I will. By the time I will be ready. Okay. Thank you.
Fine, sir. Fine, sir. A very good afternoon to everyone present here. Myself, Dr. S. Nagamani. So sorry for the delay, and due to some technical issue, we couldn't start on 2:30. On behalf of DDH 2020 training team and chairman of our committee, Dr. G. N. Sastry, once again we welcome you all for the vertical 1 session. So today the vertical 1 will be delivered by Dr. M. Karthikeyan from National Chemical Laboratory, Pune. Dr. Karthikeyan is a senior principal scientist in the Chemical Engineering and Process Development Division at NCL Pune, and he holds PhD in Organic Chemistry and working in the area of chemoinformatics and its application in drug discovery. And currently, his group is mainly working on the applying machine learning and artificial intelligence in chemoinformatics and drug discovery. And he is also the recipient of prestigious Boy Scout Award by Department of Science and Technology and Overseas Associate Award by Department of Biotechnology. With this small introduction, I request Dr. Karthikeyan to deliver the talk on remote computing using email platform. Over to you, sir.
Good afternoon, everyone. First of all, I'd like to thank Dr. Narayana Sastry and the entire Drug Discovery Hackathon team for giving me an opportunity to present our work in this cheminformatics for drug discovery. So first of all, I'd like to congratulate all of you for joining this particular event and presentations. I have seen in this hackathon, several eminent speakers spoke about drug discovery. So I don't want to repeat any of the things already spoken. They spoke about chemoinformatics, they spoke about bioinformatics, they spoke about drug discovery as a whole. Now, this hackathon may lead to inspiring students and people who are watching this program to develop some skills in drug design, especially in COVID situation. So there is a need for development of drugs. Of course, we are working towards vaccines, but drug is very, very important. Especially in drug discovery, it's very expensive, time-consuming process. So we need to develop high-performance computing enabled softwares, and it should be maybe open source. And India is very good with brain power. So I think this hackathon will help inspiring some young generation who learned chemistry, who learned biology to do some coding so that they can develop some software which can lead to great inventions. So today we are talking too much about artificial intelligence, big data, IoT. These are all the new technologies. Next 5 years, it is going to rule the world. So are we prepared for this? So I myself, as Nagamani told us that I trained in organic chemistry. I've done PhD in organic chemistry. Then along the way, I moved the field to cheminformatics. Now I am slowly moving to artificial intelligence and Internet of Things, robotics, etc. This presentation, I am going to cover two topics. One, how to do remote computing using email platform. That means you are just sending an email with a subject line containing maybe two terms. One could be a drug, another could be a target. So given two informations by text, that is, say for example, aspirin, and a protein target could be 1 HIV. Given these only two terms in the subject line of a in the email platform from your mobile, from your laptop, from your desktop, wherever, you just send an email with subject line aspirin, a drug, and a protein target 1 HIV. So imagine there is a robot which reads all the mails received by the system, and you send a specific email address. Say, for example, COAVID-TK@gmail.com. It can be any email. So you send an email to a email server with the common email ID where it is supposed to do, a robot is supposed to read your email, understand the email, understand what you are looking for, so that it can do the task. What you are expecting given ligand and target. So you want to do some docking studies. So people who learned this who learned from this all this series of lectures with respect to drug discovery, they know that docking is very, very important in drug design, because we want to know how the small molecule interact with the biological systems in your body. So in silico, how the molecules go and bind in the binding pocket. So is it a simple problem? No. So people use it, several commercial and open source softwares for docking studies. But here, how we can use email as a platform to do the docking studies, given aspirin as a input and 1 HIV as another target input. So we expect the robot should understand aspirin as a molecule and 1 HIV as the target. So it is supposed to convert this aspirin into a molecule, that means machine understandable format, atoms and bonds. And then it should understand 1 HIV as a protease. It has to download from PDB server, and then it should do docking studies and send you the binding energy. It looks like a magic. Yes, it looks a fantastic. It is a magic. It looks like a Steven Spielberg movie, but that's what we achieved. Because the science is advancing so much, then we have to use all the technologies. One person can send, as the picture shows, one person can send many emails to that email server, and everybody can send, one email. Means there is no limitations. So because you know that Google handles millions of mails daily. So we send the mail, then we need a platform. Actually, I used the Google itself as a platform to demonstrate this. Okay, so now I will go into details. So my presentation will be two parts. One, how to use remote computing using email platform, and then how to apply this one of the technologies, especially docking studies, in one of the software development. So COAVID-TK. What is COAVID-TK? It's a COVID antiviral discovery toolkit. Okay, so in this we are working towards integrated platform, which includes AI, high performance computing, GPU computing, data mining, big data analytics, machine learning, deep learning, bioinformatics. And you can do patent mining, literature mining, structure activity relationship studies, molecular dynamics, molecular mining, molecular docking, QSAR, QSPR, and QSTR. These are all structure activity, property, and toxicity relationship. So, and then you can do chemoinformatics tasks, and then text mining. Basically, what we are looking for, given COVID virus, and then the related proteases, can we find the small molecule which goes and binds, so that we got the antiviral molecule using this platform. So here I will show only one component, that is molecular docking, as an example, how sending email to the server get the binding energy by email back to you. That is what one of the technologies integrated here. So I will go into the details of this COAVID-TK software eventually. But my main aim is all the technologies can be achieved through, if you can do docking, you can do anything with email server once you have the protocols. So now I am actually inspired by this computer chemistry. That is since a long time. Since I've done PhD in chemistry, I thought that how computer can help to identify molecule of relevance and how to synthesize. What is the starting molecules for that molecule of interest? Suppose if you do the drug discovery, you identified a molecule in silico. Now you want to know how to make it shorter, how to make it high yield, how to make it environmental friendly, how to make it non-toxic routes, and then how to make it economically viable. So these are so many parameters are there once your molecule is identified. Even in our case, if we want to discover an antiviral compound, so we want to know what are the routes in which the molecules can be synthesized. Since chemistry is almost 200 years old, all data is already indexed, available in some web servers. Say, for example, American Chemical Society Chemical Abstract Service indexes each and every research paper published. So can we harvest some information from scientific literature so that it can be used for training the machine to know how to make the compound, how much yield I am likely to get given the route. So these are the things right now people are experimenting. So AI is now no longer a technologist terminology. Everybody now has to understand. Alexa, can you tell me how to make this molecule? Alexa, what will be the expected yield? This is the question people are going to pose in future. So Alexa is already there. Siri is there. Another Microsoft also developing voice-based interactions. In future, people are talking about brain-computer interface. That means you think it should be done. So people speak, people type. Now people should think. So I am going to that details also. Are we ready for this? So if I think about a molecule, I want to know how to make it, what is the expected yield, etc. So can AI play an important role? I am just going to cover up all these things in this presentation. Okay. So now these are the few marketing or people talk about AI for coronavirus. This is news articles everywhere you can see deep learning applied to drug discovery and drug repurposing. These are all not published in simple journals. These are all published in very high impact factor journals like Nature, Science, and other important journals. So that means people already achieved some level of role of AI in drug discovery. Now you can see now people want to use existing drugs as a repurposed drug. So can we use existing drugs for antiviral activity and especially COVID, because there is an urgency. So that is the easiest way because the drug is already approved, all the toxicity profile, all the efficacy, everything is studied. AI can tell whether the compound already in public domain or made by pharmaceutical industries available, and whether it is toxic, whether it is antiviral, and if it is antiviral, whether it is COVID specific. So can I use AI for this? So you can see these news articles. Everybody is talking about role of AI in COVID antiviral drug discovery. So now I will go into details how the COAVID-TK will help. So historically we use open source softwares. We developed large scale literature mining, text mining, because in text mining, given abstract which discusses about the molecule which is specific to particular disease, which is specific to particular viruses, which is specific to bacteria, fungus, etc. That information is available in textual form. Then we have to use some machine learning, deep learning, artificial intelligence techniques to translate the text into a molecule, and then understand the once the molecule is recognized, then you have to find out proteins, you have to find the disease, you have to find the organism, all the information needs to be extracted. Then manually it is impossible to harvest millions of literature articles, then we need machine help. So machine will read the word by word, all the abstracts are text, and then classify whether the text available in that particular abstract is a chemical, whether it is a disease, whether it is an organism, etc. Once it understood that, then it can build the network, biological network we call. In that biological network, what happens? Molecule associated with diseases, the disease associated with organism, etc. is mapped. Now you know the specificity of the molecule with respect to disease of interest. So this can be done at the first level in text mining. So you need molecular mining. Once you identify the molecule, then you understand what is a common scaffold. So what is a common suppose for example, chlorobenzene, where benzene is the scaffold and chlorine is a functional group. So given millions of compounds, we need to identify what is a common scaffold. There may be a benzenoid compound containing, there are about 3 million compounds you got, out of which there is one common compound, that is benzene. So now we have to identify that how many diverse molecular scaffolds are there. Then given the scaffold, why you need scaffold? Once you have the good scaffold, you can take a patent. Pharma companies, they actually take the patent on scaffolds. The scaffold is very, very important. Then the variation of scaffold and variation of functional group is very important for building the library of compounds. So then once you recognize the scaffold in a molecule, then you can build virtual library. Once you have the virtual library of molecules, which includes known and unknown compounds. So once you recognize the molecules, you recognize the scaffold, then you build the virtual library. Now you have to do structure activity relationship. That comes under QSAR. I think Professor Roy discussed about structure activity relationship. Dr. Bharatam discussed about pharmacophore informatics. So all these things are already discussed very well. So what I will discuss here, how to extract the information from literature, because we need a robotic way of reading the literature, because manually it is impossible to read 40,000 literature with respect to COVID. So now we got the 40,000 literature which is available in public domain, put by Chemical Abstract Service, PubMed. Many people are working towards putting the literature into the public domain. Now who is going to read it? So if you develop a robot which can read word by word, character by character, recognize the molecule, then you got a molecular repository with respect to antivirals. Now the chem informatics part comes into picture, because once you have the collection of molecules, then you can do all the structure activity relationship, data mining, and then other related target related studies. Then you can integrate the molecular information with the bioinformatics. Then you can do drug discovery effectively. Okay. Then vaccine design is another angle. So I will not touch into vaccine part. I will just discuss about how to design an antiviral compound using these robotic and remote computing techniques. And then whether it is how to make it COVID specific using AI, where machine learns whether it is COVID specific or not. So I will talk only about that. So we did the text mining. This particular toolkit COAVID-TK is integrated with text mining tools, molecular mining tools, and then a bit of bioinformatics tools. So this I already discussed. So now you also want, what is the role of this patient data? So we need to get the patient data. So that means this drug discovery should tightly integrated with what is happening in the hospital level. So what are the symptoms? What is the role of drugs with respect to the patient? What type of patient? What type of symptoms? All the things can be extracted. Then this will help to identify the drug which is more potent. Second, it has a minimum side effects. Okay, so for this purpose, we need to collect some information which may not be available in the scientific literature. This also comes under pharmacovigilance context, because sometimes introducing a drug and withdrawing from the market is a huge cost and loss of lives. So can we generate a AI system which creates the alert that the drug which is introduced should not be withdrawn? So can we design that type of drugs, where what is the role of AI? So this is one of the aspects usually people don't cover in drug discovery pipeline. Now this includes a text mining component. What happens in text mining? Once you given a PubMed abstract, it understands each and every word. Once you understand the word, what is the term? For example, here coronavirus, we know that it's an organism, and then what type of things? Infection, then patient, then respiratory. So these are the terms are extracted. This become input for your AI system. Then it also understands all the drugs cited with respect to antivirals. Nelfinavir, Ostelmervir, etc. Similarly, protein targets. So there are some people, some publications discuss about HIV, ACE2, acetylcholinesterase, or angiotensin enzyme receptor. So there are so many things, etc. So there are so many targets are there. Now you got the molecule. This is our first level of extraction information. So you got the high frequent targets and high frequent molecules. These are all known molecules. Now the question is, once you recognize the molecules as a drug, and once you recognize the targets, can we get the associated proteins, etc. for docking studies? Can we get the molecular structures in a truly computable format that can be used for drug design? Okay, so now this is the text mining. Once you do text mining, you got the word collection. From word collection, you got two very critical parameter, that is the drugs and targets. Now let us go into the details. So then you have so many abstracts. In one abstract, there were several terms are there. And then what is the term, which class it belongs to? Now we are looking into this text mining is not perfect. So we have to develop, still there is a scope and opportunity for developing a fantastic text mining tool, because this text mining actually misclassifies. Sometimes people use text mining based on known terms, dictionary of terms, etc. But sometimes they use machine learning to classify a term as a drug, as a target, as an organism, etc. So now you got the most frequent terms, and then you know which class it belongs to. Okay. Now you got the list of drugs. Now that cheminformatics component come into picture. You recognize the term. Now the point is, from given name, how to get the chemical structure. Chemical structure is the international language of chemistry. You may have many names for the same chemical. Usually people don't understand that. They have so many names, but all refers to one molecule. There is a trade name, there is a CAS name, there is a IUPAC name, there is a common name, there is a name given by the pharmaceutical companies. So all refers to one molecule. What are the advantages of cheminformatics? Cheminformatics actually convert all names into a single structure. Once you have a structure, once you have a chemical structure, proper chemical structure, converting all the names into a single chemical structure, then you have atoms and bonds, which is unique. Whether you have stereochemistry, what is the scaffold? In last time I mentioned that scaffold. In this particular case, you can see this is the scaffold, and these are all the functional groups, which is connected by a single bond. So our way of recognizing the scaffolds are different. This is a functional group. You can see three functional group here, 1, 2, and 3. What about this? This entire any ring containing compound and the ring compound which is connected by atoms are also part of the scaffold. So when we do the scaffold recognition, we only disconnect the atoms or functional groups connected by a single bond. So in this case, you get 1, 2, 3, 4, and 5 functional groups and one scaffold. So this is the way the machine should recognize a scaffold and a functional group. In so many commercial softwares, people say that no, my scaffold is only this, and this is also a functional group. So it is a different way of classifying scaffolds. Okay, so now this software is capable of recognizing the scaffolds, functional groups. Why we need this? In future, we want to generate a new molecule from the known molecule. That's why you come into invention. Then you come to discovery. You can have a patentable molecule which is not yet reported. How to generate a new molecule from known molecule? This is the strategy where you recognize the scaffold, recognize the functional groups, and use combinatorial methods to regenerate all possible combination of focused virtual library. That means only this atom is connected to particular functional group, so that your new functional group will come only here, which is similar to this carboxylic acids. Ester can come, functionalized esters can come and attach this particular atom. Similarly, you know that this particular functional group attached to nitrogen, only the similar compound, similar functional groups can come here. Here you see oxygen, so similar pharmacophore based functional groups can come here. So it is not, people do different type of combinatorial library. This is a focused virtual library where R1, R2, R3, R4, R5 are replaced with similar functional group. Here chlorine is there. for example. All halogens can be replaced in this place only. No other functional group can come here. So you can restrict your virtual library generation using this. So you may be thinking, what is the use of this? So if you tell computer that these are my known compound known to be biologically active, the computer is supposed to generate new compound with more efficacy, better results, minimum toxicity. That is where this cheminformatics plays an important role in drug discovery. So then what is the next step? So you got the molecule, computer don't understand the structure, computer don't understand the picture, computer understand the beautiful fonts which you have drawn. It doesn't understand the three-dimensional images which you show to the computer. Okay, so now you have to translate this molecular structure into numeric form because computer understands only 0 and 1. Computer only understands the numeric for correlations. So how to translate this pictorial information? First, it was a textual information which has no meaning until unless you convert it into a molecule. Once you have a molecule, you have atoms and bonds. Then this becomes very, very important. From this molecular information, now you are going to generate lot of molecular descriptors. You are describing the molecule. What is the characteristics of that particular molecule? Whether it is a polar, whether it is aromatic, whether it is soluble. Because you see that log P. What is log P? Octanol water partition coefficient. What is the solubility of this particular molecule in water with respect to organic compound is a parameter. So can I calculate that value based on known log P values of series of compounds? So this prediction calculations all are done by commercial software, open source software. Now you translated your molecular structures into numeric form. Now it is ready for any type of drug discovery pipeline. For example, first time I want to thank American Chemical Society and especially CAS for releasing 50,000 compounds. This is the first time I want to congratulate them. I want to thank them because that is the first time for the society they released 50,000 molecules with respect to antivirals in the public domain. Usually CAS doesn't release even more than 100 compounds for a licensed user. And for a commercial users, for a pharmaceutical companies, the licenses will be different. So they will be eligible to download up to 500 compounds. You can see that because of the COVID pandemic, all coming together to help each other. Now they released 50,000 compounds. So once you have 50,000 compounds extracted from CAS literature, now our job is to find what are the unique 23,733 molecules. Because these 50,000 may have a lot of duplicates, a lot of mixtures. So we need to identify unique molecules using cheminformatics tools where we got now 23,733. Okay, once you have the library of compounds, now you have to find out where this 23,733 compounds stands in the chemical space. So are they all similar? How the property space they vary? What is the commonality among themselves in terms of structure, in terms of functional group, in terms of polarity, in terms of energy, etc., etc., etc. So these are actually called molecular parameters, molecular descriptors. Now you in the descriptor space you are defining the library of compounds. So now you know this particular feature. These descriptors are plotted for a series of compounds. Okay. Now you know what range they come into. Particular descriptor, which range it comes to. So let me show you some example here. Say for example here, S log P, you should be for a series of compound, it must be below 60, 0 to 60. So you cannot see that there is no molecules in this range. Among this, the actual compound, actual antiviral compound, if you want, you have to look into 25 plus or minus 5. So even though you have a library of compounds, but maximum compounds comes under this range, and the dot says that what is the actual value? 25 is the actual value, S log P. If the molecule is 25 plus or minus 1 or 2, could be a antiviral drug. Because anything, then you can use this one as a filter. So you can say that these are all now become filters. These are all you can use as a filter. What parameter B cut should be 2.75 to 2.5. So this manually it is impossible. That's why we need machine to generate, machine to understand, machine to develop a model, and then machine to screen the compounds which is of your interest. If you're a very good medicinal chemist, you can design a compound by paper and pencil. Now, then you don't know beyond that. You know that this compound is likely to work. Now, given that particular molecule into the system, like this type of system, where you can get the whether it belongs to wherever the black dot is there, whether the molecule which you are thinking is comes under this Lipinski rule of 5 is just 4 parameters. So now you can see so many parameters are there with respect to antivirals. So you can also build your own descriptors. So this now all I want to tell is that given name to structure, structure to numbers from series of compounds, now you get the collection of compound. What is the range in which these compounds fall into? Why this range is important? These are all antivirals. Now first we need to know what are the ranges for antivirals and what is the range for COVID specific antivirals. So now this is the first level of screening. What is the second level of screening? Among this, which one is supposed to be COVID specific? Okay, so now I will go into the further details. Okay, so now you have millions of compounds out there in public domains. Okay, I will show you that list also. You have a huge collection of antivirals, antibacterial, natural products, synthetic compounds. Everything is available in SMILES format, SD format, commercially purchasable compounds, etc. are there. Now your aim is, can I filter some compounds which I want to buy and I want to test against the my targets of interest. So one is in-silico screening will help you which molecule to buy. In-silico screening, suppose 1,000 compounds are sold, 50,000 compounds are sold as a library. Now you can tell the vendor, no, I don't want 50,000 compound, I want only 500 compound because I don't have money, but I want the proper 500. Which 500? You can select based on these models in the descriptor space, based on the potential, since you already built the ranges, using this range as a filter, now you pass the 50,000 compound, come up with the top 500 compound which meets your requirement. Now you can reduce the cost. Now these compounds are likely to be biologically active because it passed through certain cheminformatics studies. So now once you have the hits, which is 50,000 to 500 you got, now you can again further do toxicity studies, activity studies, etc., etc. Now you can further filter set of compound. This is the main aim of hackathon. Why we need? First, you have so much information is there. Can we use technology to filter the compound, come up with 2-3 compounds which can be tested in the laboratory level? You cannot test millions and billions of compounds in the practical way in the laboratory environment. So you do some filter mechanism. Now that's what all people talked about so far till date in the hackathon. So now you got a very clear picture of where the molecule is coming from, how it is being calculated, how it is being shortlisted. Then you can do normal pipeline, etc. So we want to do it very simple way. So because we know that chemists are not computer savvy, they don't do programming much. So we want to help the chemists and biologists who are not much expertise in computer programming. So can we build something which is meeting the requirement of both chemist, biologist, etc. So then we want to make a pipeline. So all they know is, okay, these are the list of compounds. These are the list of biological activity with respect to the compound. I have a table containing name of the compound or identifier. Maybe I have a structure which I don't want to disclose. And then I have a bioactivity which I publish in the journal. That's all they have. Given that as an input, I want a new molecule which is not yet reported with potential bioactivity. What is the procedure? So you take that 50 molecules and 50 bioactivity which is reported in Journal of Organic Chemistry, Journal of Medicinal Chemistry, any journal, and input to the systems. Just if generally what happens in journals, they don't release the molecular structures. They give the chemical name, they give the CAS registry number, etc. But they don't give truly computable molecule in SMILES format, SD format, etc. So the problem for the chemist is now After reading the paper, they have to painstakingly draw the molecules and then verify the errors in the structures, and then again they come into cheminformatics. So in that process, they lost the interest. They no longer do the QSAR. So then somebody who actually interns, they actually draw the structures, then they release the molecular structures as a library. Then someone else take the things, and then they process the things, finally come up with good QSAR models. So if you see people who are working on QSAR, they don't do the experiments. They depend on experimental done by the biologist. So now any error introduced in the experimental, in the design, all leads to problems. So error reduction is very, very important. Then only machine will work. If you introduce the error and noise in the system, then basically what you get is a noise. So if you remove all the noise in the systems, you will get a good models. So in these particular things, I want to explain. You give the molecules and bioactivity, then you calculate the descriptor automatically. Then because the number of molecules and the targets are huge, then you need GPU performance, 50,000 core, 70,000 core, 100 servers. So all work in parallel, in a distributed manner. Because nowadays people talk about machine learning, deep learning, but they forgot the computing power required for performing the deep learning techniques. So there are so many open source deep learning and machine learning techniques are available, and now there is a complex problem involved here. One, the data is huge, and the computing requirement is also huge. Then storage is also huge, because your input is only 50 molecules, but when you process all the things, you will end up maybe with a few gigabytes and terabytes of data. So now machine learning and deep learning leads to building mathematical models, which you can explainable models or non-explainable models. Now people are talking about explainable AI, interpretable artificial intelligence. Explainable artificial intelligence. What is explainable artificial intelligence? They are actually mathematical models. You can explain what is the parameter responsible for bioactivity. So the model should explain these are the parameters. If it is there in the molecule, it will be definitely biologically active. That is what they are looking for. Now, in a conventional way, you have a model, you have a default model, you have a free filtered model, you have an accurate model. Then you validate the model with external data. What is external data? External data is the one which you did not give the computer for training. Internal data is the one actually this biological activity. Suppose you have 100 molecules and corresponding bioactivity, you give only 60 molecule as a training set, and then you build the model. Remaining 40 you never gave to the computer, because you know the bioactivity, you know the molecule for 40 molecules. Now given 40 molecules, can you predict? So observed versus predicted. So if they all stay in the same line, then you can say your model is good, but it never be. So there are some outliers. What is predicted may not be exactly observed. So in your 40 molecules, you will find a lot of outliers which is not falling into the trend. But then you have to compromise. Okay, either use the model or don't use the model, or use the best model. What people do conveniently, they remove these outliers and try to make a best model and publish. That is wrong. So what happened? That will work only for the 50 molecule. It will never work for 51st molecule. So this is also a problem in structure activity relationship. Basically, you have to tell the computer the truth. What is the molecule? What is the bioactivity? What is the diversity? What is the variation? Everything should be given to the computer. Then only computer understands what is the error, why it went wrong, why it is outlier. Can you explain the outlier? Why it's supposed to be here? Why it came here? So those things can be done using AI techniques. Now you got the predicted bioactivity. You save the model. Okay, so that completes the complete cycle of structure activity relationship. Once your models are ready, you take a virtual molecule or commercially available compound, then process them, and you have a predicted bioactivity, IC50, GA50, EC50, IC means LD50, lethal dose, anything. So basically, you will be doing different bioactivity, different models, and for the same compound, you will apply different models. Then you predict activity, property, and toxicity. Three things you predict. Okay, so once you have the predicted values, based on your expectations and requirement, you can shortlist the compound which is of your interest. Okay, that concludes the first part of the presentation, where given name and bioactivity, you got the model and you applied the model on virtual molecule or commercially available compound. Now you got the predicted bioactivity. Now let us go to the second part. Once you know what we want to do, okay, I am not interested in all these complex things because I don't know. All I know is these are the drugs actually used in the clinical trials. These are the clinical drugs because it's gone through all the process. Now I have some 3000 drugs. I know these are all drugs. I don't want to work with millions and billions of molecules which computational scientists work. I am interested in only drugs, clinical drugs. It can be antivirals, antibacterial, anti-cancer, anything. I want to take a selected set of clinical drugs. Now I want to do scaffold analysis. What is the common scaffold among these particular clinical drugs? Once you have the set of scaffolds and functional group with respect to the class of the particular drugs, then I can build the virtual library. You already built the structure activity relationship for all the drugs. Now you generated the new molecule around those drugs automatically, not manually. Only medicinal chemist can build the molecule manually. Otherwise, it is impossible for building a new molecule from the known molecule. So then computer actually recognizes the scaffold, recognizes the functional group, and then it built the virtual library of novel molecule. Because in a research, we are looking for something new, something novel. Then only people will appreciate your efforts. So you got the new molecules, apply the models which you have done on drugs, and then go through the same pipeline, compute descriptor, use models and predict bioactivity. Now what you got is drug-like compounds which you generated for the particular class of disease or the particular disease or organism specific. Now you got the focused virtual library with respect to the drugs which you have taken. If you are taking fapiravir, all type of XYZVIR compounds. Now naturally you will end up with vir end type compounds. So you got a focused virtual library of antiviral compounds. Now antiviral compounds, new antiviral compound can go through this pipeline, but there is a risk. Who is going to synthesize? Who is going to biologically evaluate your new molecules until unless you give the confidence? So right now people are working into drug repurposing. Once that story is over, now people will go to novel molecule. This should continue. This study will continue. So the machine will learn using machine learning, deep learning, virtual screening. Finally, it will understand and finally it will give a bioactivity for the known compound. That means this particular predicted bioactivity will be really good, expected to be good. Okay. Now, again, chemists and biologists don't do programming. So now you have a software, commercial software, open source software. There are so many buttons, so many commands, so many menus, so many parameters which you have to fix. So it is very, very complex for people who want to try out something. So what we are trying is, given a list of name of the chemical or a structure or target, I want the binding affinity for that compound using a standard operating protocols. Okay, so now it is very difficult, and I'm telling you, even though it is difficult but not impossible. So you need a huge computing resource, maybe in a remote location. Why we need to do docking? Because your small molecule interact with the receptor, which is giving some response to the disease of interest. So what is happening in biological system now, using In-Silico, you identify the targets of interest. In our case, in COVID, we are talking about spike protein. There are several type of proteins with respect to COVID. So you take those targets and you already know all the antivirals and antiviral like compounds, and then you want to give as a automation method, so that known compound is docked against known targets. So you have a library of target with combinatorial docking studies, and the results are now in a matrix form you have available. So now you can compare that with the new molecules which you generated using virtual library screening. Now this is just a small slide. Actually, I borrowed this from Chemaxo. Chemaxo is our partner. And the last 15 years, I am using Chemaxo. 2007 onwards, we use only Chemaxo tools because previously they used to give free. Okay, so unlimited licenses. It was fantastic software. The good thing about Chemaxo is you can actually use the code inside your program. Okay, so that is called API, Application Programmable Interface, so that you can use the code. How the Chemaxo works inside, you can use it. So why we need Chemaxo? To handle huge data, millions of data, you need tools, open source tools, plus open source databases and academic tools like Chemaxo. Chemaxo now it is reaching all the pharmaceutical industries because you see the level of molecules which we are talking about. So this is a Molport is a drug, means a commercially saleable. You can actually buy 28.3 million compounds. You can select the compound of your interest. It is purchasable chemical. People talk about so many other libraries, but these are all the very important. Why Chembal? Chembal as the molecule with bioactivity or relate, maybe they won't give actual bioactivity in nanomolar or picomolar value, but they can give this. The compound is annotated with antiviral, antibacterial, natural product, synthetic. Those type of information also very important for machine learning and deep learning. Binding DB, which tells the how the targets are, how target and ligand interactions. So if you want to know what is the active site for a molecule, you can find the native ligand and then you can use one of the anchor where you can your molecule can go and bind in the native ligand space. You can also identify different active sites using several methods, inbuilt methods available in your open source and commercial softwares. So identifying a binding site where to bind is also very important. So binding DB, Chembal, all the tools are now integrated with our software. Okay, we have some huge collection of all known compounds available in public domain. So in these particular studies, what we are doing is take some 118 antivirals and then do the automated docking and QSAR related studies. So this one particular slide shows where is the white space where you can see a patentable new molecule space. Wherever the white space is there, that means molecules are not explored in the diversity space. So these are all diverse molecules. So you can see that in this space, a lot of molecules are processed with the context of antivirals. You can also look into nearby space. Okay, spear exclusion techniques people use to identify what are the similar related compounds which is not yet reported. Okay, and likely to have the same bioactivity studies. So you can take the targets, you can do the drug studies, you can do the binding studies, you can do structure activity relationship studies, you can do molecular diversity and similarity studies. So this all integrated in a single click of button. Okay, and then you compare your automated techniques with commercial softwares, how good it is with respect to Glide software, with respect to MOE software, whether I'm getting the same binding pocket, whether I'm same binding energy, how it is variation. So with the solvent, without solvent, with water molecules, so etc. with metal inside. So whether all the information now you can do comparative studies. First you do automation, that means you end up with small molecule and confirmation. Now you compare that with commercial software to validate your own technique. Now, once your results are comparable with commercial and what you did with open source, then now you can trust your open source, because what you do large scale screening may not be possible with existing licensing, existing software specifications and existing hardware. So now you learn, you can use this as a training set and then compare with what you got it from your this open source design, etc. So you can see the results. Okay, variation. Now I am almost close to the end of this particular session of presentation. Basically, you take the information from different sources, then you integrate them, identify the molecular networks. That means scaffold analytics, which is not available in any of the software today, I can tell you. Okay, so you need to identify the proper scaffolds which can be used for virtual library building. So there are bits and pieces available in isolation, scaffold identification in some software, and then virtual library in another software. So this is what I discussed here is not available in any of the software. Okay, so I can now skip. Okay, FDA discussed, FDA approved drugs, antivirals I already discussed. Now what is required is how to use AI for this thing. Now I will focus only on that main theme of one of the module. I will discuss only one module. There are several modules which I discussed, but only one module I am going to focus on. That is how to compute using remote method using email as a platform. Okay, this looks like a magic. Please pay attention, and it will be very interesting for you. It may instigate you to do more research. Okay, so what we did, So since I am today, you can see our the pulse of the generation is people with chemistry can do computer programming skills as per our leaders are now talking about. So there is a multidisciplinary is very important. So don't shy away from computer programming if you are good in chemistry. So if a person is good in chemistry, basically is very good in logic. So since logic is very much required for computer programming, you will be the best person to do the computer programming with chemistry knowledge. Now you apply these two together, then you become a chem informatics. So that is how I evolved last 30 years. I did PhD in organic chemistry, but then eventually I become chem informatician because I got inspired by leaders like Professor Johnny Gasteger, Alex Tropshaw, etc. So these people actually did chemistry, and then eventually they moved to chem informatics. Then you can do better, tool development. So then I believe in open source tools because it is easy for me to distribute after publications. That's why we focus on open source tools. Now, artificial intelligence, machine learning, it is not actually new, but eventually getting popularity because of the complexity of the data, etc. Then you can learn from academic and commercial software. Academic is slightly less costly than commercial softwares. So then it will be better if you do it yourself. Because when you do your own software, even small Python programming, Java programming, C programming, whatever you develop, and then use that programming to connect to a database, and then slowly learn name putting into the database. Slowly you can start with. Eventually you will be surprised to see how big the software is developed. And then the good thing about computer software programming is you can reuse it, your own code again and again as and when required. So you develop a code such that you can use it forever. You don't need to modify the modules. You keep on generating small module, but when you integrate all of them, it looks like a very big device. Okay. So now I want to just, this is what I work with: Java, Python, C++, and I use NetBeans as an IDE platform. And then I little bit learned about networking. And then MySQL, PostgreSQL, these are all the free softwares. And PostgreSQL is like Oracle software. It's a huge, it can handle huge data. And now people are talking about NoSQL. So that means you don't need the standard database system. So you can use data as a file. Okay, that will be very fast, extremely good, easy to distribute, and it will be very fun, useful for distributed computing purpose. Okay, so now I am going to focus on. You can see that I'm going to show how our email system works. Basically, what you want. So you send one email to a website or a email system. Here, what I am sending is COAVID-TK @gmail.com, where I want the results. Okay, so now I will go into the details how it is happening. Okay, just to send the subject line with the two terms. One is a drug name, another is a target. Okay, the email reading robot which developed using Java platform automatically retrieve the email every minute. Okay, every minute whatever the email sent to that system is read every minute, once in a minute. Then it understands the subject line, understand whether it is a name of a chemical, whether it is a drug, etc., target. Then it performs the pipeline process which I explained to you, how the docking is done. I will show you also here. Okay, before this presentation, I was little bit had difficulty because of networking problem. Actually, I tested before giving this presentation whether it works. Okay, 5 minutes back before the presentation, actually I sent the email. I got the results. I will show you. Okay, so now this is what we filed a patent because nobody did. This is one example. We did barcoding, large scale barcoding, large scale virtual library in a barcode. Now we did that type of information passed through this email system. Suppose you send a barcode and you want a virtual library to be screened. This can be done. So we filed 2 patents with respect to these studies. Okay. Now, how it happens? You can send email from any technology, whether it's a mobile phone or a landline, whatever connections. Once you send the email using to a system, then it checks validity of your email, whether you are a registered user or not. Right now, we have freed the system for testing purpose. Anybody can send from any corner of the world. So the mail is read. Okay. We actually tested with several modules. Only I will be talking only about the molecular docking with, say for example, aspirin 10GS. It is a target name and this is the drug name. And then once it is sent, then the program will do the docking studies. Now I will skip some of the slides because of the lack of time. This is virtual library design. Basically, you send some set of compounds as an input, then you can get huge collection of virtual library, maybe a barcode as an output. Okay, so now what happens? So you send the data, the robot reads, understands, and then it executes a set of sequence of tasks, and then it produces the output and send it back to you. So you can see that this is on 17th July I tested when we finalized this recently, again for this demonstration purpose. Then you send to COAVID-TK, then it was sent. Okay, this is only text you have to type, one HIV space paracetamol. That's it. Now you will get this as a input is the 2 terms and your output is one HIV_paracetamol.zip. So what it contains, the zip file containing all these files, it is automatically generated by the system. It converts your name to structure in SMILES format. Then once it has SMILES format, it converts SMILES into mol2 format, PDB format. This is the ligand directory. So it converts your paracetamol into mol format, mol2 format in 3D space, and then it can optimize the molecular structures. Then it do energy minimization. Then you got the optimized energy, and then it generates the PDB-QT file automatically using some ligand preparation scripts. And then it also prepares it download the one HIV from PDB site, and then it will do the processing. Finally, you will get the ready to dock target and ready to dock ligand based on your terms which you submitted. Okay. All the procedures are mentioned in the patent. Now, then I requested people, can you test the system? I'm keeping my laptop on. Can you send? I called all the people. Okay, it came from different. You might be recognizing some of the email IDs. Giri, Amit Saxena from CDAC. So Somasundaram from Naipur, Kolkata. So different part of the country, different organization, different backgrounds. I requested them, can you send some name and target? I requested them, any of your imagination, whether the system works or not. So then I gave example, one HIV and aspirin as an example. But you can see that people tried with the metformin, so many things they tried. Okay. Now what time it was received, and then the result send it to those people, all these people. Okay. Now these particular things, August 13, 14 hours, I sent an email to the system from my email ID. Okay. Now it sent the results back. I tested it. Okay. So basically, you can send. One day I will open it, provided the Gmail don't have any objection. So once you have millions of mails, all will be processed using this, and you will end up getting these output files as a docking. Same thing can be repeated for a set of compounds. Suppose you want to given set of names, say 50 names I sent as attachment. Can I get all the predicted bioactivity in this system, antiviral activity, anti-cancer activity, etc., etc. Now, since we demonstrated this, now everything is possible, right? So it is up to your imagination. Basically, send the email. Your systems with good computing infrastructure should be there. All the open source softwares can be used. You cannot use commercial software for this type of studies. Okay, so you have to be very cautious what you use, where you use, how you use. There are some limitations. So if you are building a system, you have to think about open source, then there is no restrictions. So it comes through the system. You have no programming restrictions. Then you got the data. Okay, so now your databases can be built. Now, one HIV aspirin as the input, then it goes through name to structure, 2D to 3D, target identification, whether available in PDB, download from the PDB, then it prepare the target, it prepare the ligand, and then it does the docking studies, and then it gives you the poses and values. Okay, so is it working now? Yes, you can say that 5 minutes before the presentation, actually I tested because I want to be very confident what I am presenting. Okay. I have to thank Chemaxo because that is their core technologies are really helpful. Last 15 years, I am very conversant with protocols, licensing, modules available in Chemaxo. So if you want to do substructure search, exact structure, similar structure search, millions of compounds in the fastest way, this does very fast. It can handle huge collection of library of compounds. There is no restrictions. Okay, it can do all type of file format conversion, which I discussed. Then it converts chemical terms, documentation library is there. Accurately it predicts, it generates the 3D structures or 2D structures from the chemical name. So we use this for the fast things. We have some collaboration with Chemaxo, so there is no restriction for using for our research. So for distribution purpose, then I have to see the licensing policies. But internally using research purpose, they are very generous. Okay, so then visual computing. Now I want to, because you must be very bored of all these studies. Now I think you can recognize who is this gentleman. So long ago I saw a movie, Terminator. Okay, he is like a robot. So what he does? So he recognizes the bike. Once he see the bike, something comes as a text. Okay, so this is the image processing. I don't know whether it worked at that point of time or not. Today it is possible using OpenCV. There are artificial intelligence. People are trying to recognize the objects, and then they want the name, they want the color, etc., etc. So now this movie was almost 20 years back, or 1970s or 80s, I don't remember. So they shown that you see the things, he wanted the details, which model, Harley Davidson. Then you see, okay, this is Yamaha bike. Okay, now he want another bike. This is also Harley Davidson. Then he chooses to take, and so in our case, what we want? All we want is a drug which can cure this COVID. We want a drug which can cure cancer. Can we use this visual computing for this purpose? Can we get inspiration from here? Can we do in chemistry in real time? This is the challenge for the young generation of people who may be watching right now, or who are likely to watch this video in future in YouTube. Okay, can I recognize the molecular structure looking at, like when he can recognize the bike with the models and names? All I want is looking at the structure. Can I get the name? Can I get the bioactivity? Can I get the molecular properties just looking at the structure? Now people talked about Google Glass. If you wear Google Glass, maybe in future you will be able to do that. Then who is going to write the software? Okay. Now in this context, it was very exciting. Sometime back, I thought that if I show hand-drawn molecule on a paper, show the web camera, I want the automatic chemical structure generation, and then energy optimization. I want the property prediction. So 3D generation, name generation, and property prediction was done in 2014. So that also was filed patent, and it was granted in, it was released in, when we submitted, it was in 2014, actually 2012. And then it took almost 5 years to get the patent granted. So now we have a tool, given a molecular structure, hand-drawn structure with paper and pencil. Now you can do molecular structure generation. In future, I am talking about the people who will be watching in future, maybe 2030, 2050. I just want to imagine. I just want to dream a molecule. Okay. Can I do the docking studies? Can I do the docking studies just by thinking about a molecule? Okay. So now it is not impossible. I have seen recently 10-15 examples. They did real life examples of brain-computer interface devices that can control with your thoughts. So now my next mission would be, can I get access to these BCI devices where I just think, then docking is done, I get the binding energy on the screen. Okay, so this is what the future holds. This is for the young generation, because right now I don't have the helmet with all the sensors here. So when I think, I see the drug and target without touching anything on the screen, just imagining the molecule appeared and target appeared and went and docked, and then I got the results. Okay, so just to make this talk little bit interesting, I am giving you some thoughts for the thoughts for the young generation. Okay, so whatever I discussed, actually we published in a textbook format. It has all the open source tools, techniques, chem informatics, tools, data techniques, everything is presented. So how the software was written, what are the codes, how to write a software, read a molecule, write a molecule, build the big data, how to do machine learning, deep learning, how to do cloud computing, all the details are in a practical context. See, you can read car manual, bike manual, etc., but you will never learn how to ride a bike or car. But if you go to a driving school, okay, so you sit in the car, you drive yourself, you see all the practical things, then you come up with a good driver. So we thought that n number of theory will not help the people, and especially in India, people they avoid computer. Chemist people don't do computer programming. Then being a chemist, can I write something which chemist can understand? Okay, in a very simple form. Then we wrote all the things. Then we actually published all the things in a research journal also. I think I am almost completed my presentation. Whatever the queries you have with respect to this presentation, please shoot me an email, okay, in any format. You won't get automatic reply back, so you will get manually curated results. Okay, so I completed my presentation. If you have any specific questions, I am ready to answer.
Sir, thank you, sir. Thank you very much, sir.
So how to exit this now?
So just you have to escape.
I escape. Okay. So are you able to hear me?
Yeah.
Okay, good. Thank you so much, Nagamani and Dr. Sastri, for giving me a chance to speak. Actually, I did not give a presentation. I spoke about my passion. Okay, so is there any specific questions from the audience or people who listen to this lecture?
There are few questions. Sir, the first question is hello, sir.
Yes, I am. Can you see me, or
yeah.
I just put the video on?
Sir, actually, you have not unshared the screen.
What is that?
You have to unshare the screen, sir.
Stop sharing. Okay.
Yeah, it's fine, sir. Sir, there are few questions. So, some of the students, they are very much impressed upon your presentation, and they are asking very general question like, what type of machine learning algorithm is good for chemoinformatics analysis?
Okay. Can I take all the questions so that I will go one by one?
Sure, sir. Sure, sir. And
they may be connected.
There is, yeah. Then another question is, one student he is telling like he is from computer science background, so knowing Python is better to deal with QSAR. And,
okay, that is also a good question. Okay. The first question, machine learning algorithm is which one is good or how to identify? That is very good question. Second one is Python. Okay, computer science background. Okay. Any other question?
Yes, sir.
You tell me all the questions. I will answer in 1 minute.
Sure. And sir, one more technical question is, does that Smiles notation exactly matches a structure, or is there any ambiguity involved?
Okay, that also I will answer.
Yeah, and one more student, he is interested on your book, and he is asking what is the cost of that that book.
See, if they download for free, I get popularity. If they buy, then I get royalty. Choice is theirs.
Okay. And one more student, he is asking like regarding that email platform, what you are explaining. that what is the deal with the privacy of the data once sent to the email or email ID for the research purpose? So these are the
that is very good question. Privacy with respect to email platform. This is very fantastic,
yes.
because he must be interested. He's from pharma background, pharmaceutical company background. Okay, that's why he's asking specificity, because whether he can take a patent on the data generated by this platform. Maybe in this context, he may be asking. Okay, I have answered for that.
Yeah. So these are the questions, sir.
Okay, so now there are 4 questions. First one is which ML algorithms and how to select. Okay, so I recommend. Okay, so now machine learning is almost dead. Okay, now we have to go for deep learning. First of all, I recommend the students to go to Wikipedia, understand what is the difference between machine learning and deep learning. Machine learning is 30 years old. People talked about random forest. People talked about PLS, PCR, principal component regression, principal component. They talked about SVM, SVR, so support vector machines, etc., etc. Those days. Now, with the evolution of deep learning, the machine is learning actually from your data. It is not missing. There is no ambiguity. So everything is dynamic learning. I also mentioned a term called explainable deep learning and interpretable deep learning. For that, I recommend first you try with your machine learning techniques, any machine learning technique. Take a iris data set, put it into a many machine learning technique, take all the algorithms, you use it, all algorithms. You should not, either it's a classification or a regression, you use all of them depending upon your data set. Then you see what is the accuracy, what is the reproducibility, what is the, because this R square value, correlation coefficient, and then RMSD, root mean square deviation. These 2 parameters are very, very important to identify which algorithm is good in terms of machine learning. I'm not sure whether he understood what I am talking about. Maybe this answer may help him to go and understand this. Okay, that is the first question is answered. If they have questions, actually, you can go back to my video and see the last slide where my email ID is there. It's very simple. karthikeyan@gmail.com. You can send your questions with respect to the presentation. Okay, then if you're sending the question, then you should like that video. Second thing, okay, computer science. He has a strong background in Python. So I can tell you, this is the very good, Python is the today language of choice. So now Java integrated with Python is the best deadly combinations. Okay, then C++ programs return and exe files created. Or actually you can see in my program, Java program using Python script for molecular structure recognition. Molecular structure, especially ligand optimization, ligand preparation, everything is Python script available in Autodoc VINA format. But the Java is using that Python script to process the small molecule and target molecules. So Python is very, very useful for machine learning, deep learning, and that is the only available right now. People talk about these notebooks, Python notebook. That is also very, very important. Now, I think that question I answered. That's computer science person in collaboration with some chemist, because computer science person learning chemistry is difficult, but computer science person working with a chemist or biologist can do wonders using machine learning and deep learning, because that is what I've shown in news articles where people are doing this. You don't need to have any knowledge on chemistry. You leave the chemistry to the chemist and biological value to the biologist. Okay, so then you just being a supporting, or you become a strong person who can convert their dream into reality in the form of Python script. And if you make it open source, I think I can download and integrate and give it to public, your modules. Okay, so I think Python code also, Python programming also I explained. So third question is, what is the ambiguity with respect to smiles? Is it okay? Python is answered. Yes. Okay, yes, so now I will go into the smiles and ambiguity. So when I gave the presentation, I told there are several names for the same chemical. Then you convert it into a chemical structure, but I did not mention it has to be in smiles format only. Okay, why I did not insist that time it is one be smiles format? Because several smiles can have same structure, single structures. So that means one structure can be represented by several types of smiles. Okay, I recommend here you have to do canonicalization. So there is a term called canonicalization. You give hundreds of smiles, it converted to a single smile. Okay, that is called canonicalization. So that process is very important in chem informatics. Otherwise, you end up when you download smiles from different locations, there may be some duplicates. That is how these 50,000 compounds were converted into 27,000 compounds, which I presented in the slides. Okay. Now, I think that question is answered, yes. Nagamani.
Yes, sir.
Canonicalization. Okay.
Yeah.
The fourth question is, what about the privacy? Okay. Privacy, everybody wants privacy. They don't want to send email. If you don't send email, you won't get the results. So if you are in a corporate sector where you want to use this platform, you can do one thing. You take the open source code, put it into your email server. Your email server means you purchased your own email server. You send it to yourself in that email server. That means no third party is involved. All you need to have an email server, because now your email and the corporate email only. Suppose if I am working in NCL, I won't mind anybody reading within NCL my email because we are a family, right? Similarly, within the system, within the family, within the group of people, you create one email system because there is no manual involved here. Manual intervention is not there unless if you are taking a Gmail, Outlook mails where people can read. Why I demonstrated this? Maybe your emails are being read by Google, maybe read by any other service provider. So if you are very much privacy concerned, okay, whatever email you are going to send also may be read by Google. Okay, so I think that is a very easy to solve problem because you have your own email server. There are open source tools to build the email server also. So you can provide as a service to the people. Okay, I think that question also answered. If you have any, okay, what do you think? Is it answered?
Yeah, yes, sir. Sir, one last question. And sir, what type of algorithm that you have used for the docking to that email platform?
That is a very good question. You might have noticed that in this docking studies, when you got the results, we have not used any algorithm. I used freely available Veena docking software. Okay, Veena docking software, everybody knows, but you need several parameters in addition to molecule and target. You need binding pocket values, X, Y, Z coordinates of where the molecule is going to bind. That value need to be given. And second, you have to give the grid box. So now we created the mechanism in which these two parameters are identified from the target itself, so that it is able to do binding. That's why in the subject line, you don't need to send the. Now we modified the software where you can send a file as an attachment. Now instead of subject line, that is only for demonstration purpose. Now the attached file will be read line by line, character by character to understand exactly what you want to do in refined docking. That can be done, and any other activity can be processed using this email platform.
Thank you, sir. Thank you very much for your wonderful session, and you have practically explained how we can directly apply this machine learning as well as artificial intelligence in the drug discovery. So I hope this your session will be very helpful for the Drug Discovery 2020 participants. So on behalf of Drug Discovery Hackathon team as well as Dr. G.N. Sastry, once again, we thank you, sir.
Thank you so much for the team and all the participants who spent the time to listen to my talk. Thank you so much.
Thank you, sir.
Okay, bye.
Yeah, bye.
I can disconnect now?
Yes, sir.
Thank you.
Thank you.